Welcome to day 5. It's been a while since I posted day 4, where we reviewed some concepts about the DNA and RNA. And yes, today is day 5, and we are going to learn some fundamental concepts of the process of protein synthesis, how it is really made, the actual process. Protein synthesis always starts from the mRNA. The mRNA is made from the DNA in the nucleus. So this is the mRNA strand and this is the nucleus. When the mRNA is made, it passes through the endoplasmic reticulum to the cytoplasm. And you should remember from the cell organelles part what the endoplasmic reticulum and the cytoplasm is. To review, the endoplasmic reticulum transports RNA, proteins, or other things. It's like a transportation route for materials in the cell. And the cytoplasm is basically the jelly-like substance filled in the cell between all the organelles. So the mRNA strand moves to the cytoplasm. And this is called the transcription. Next, after the mRNA strand moves to the cytoplasm, it is broken down to small parts. This happens because some part of the mRNA strand does not contribute to protein synthesis, while some parts of the protein contributes to the process of protein production. So let's call these red mRNA parts not the parts that are not contributing to the protein synthesis, and the blue mRNA parts are contributing to the protein synthesis. These red mRNA parts are called introns, and since they are not useful, they are broken down by special enzymes, and they just disappear. However, the parts that contribute to the process of protein production is called exons, and they fuse together, they connect together, and form a new mRNA strand that is fit for the production of proteins. And now, this is called regulation. It's really basic. And you remember from the review and the concepts of RNA that these strands RNA has the base pairs of adenine, uracil, guanine, and cytosine. So yeah, they have these base pairs right here. Okay, once the exons connect together and form a new mRNA strand, it fuses again, connects again with the tRNA. If you don't remember what the tRNA is, so this is a really basic form of the tRNA, but it basically looks like this. Yeah, it looks like this. There's one amino acid per one tRNA, and it's in the top. So, okay, once the mRNA is ready for the production of proteins, tRNA appears. It's a chain, it's actually a chain, it's connected all together. The tRNA chain appears and it connects with the base pairs of the mRNA strand. And these tRNA is represented as codons. So you can call these as codons. once the right base pairs are paired with mRNA and tRNA, the amino acid that is located on the top of tRNA is separated. It's separated. And yeah, since this tRNA is paired rightly, and this is also separated, and there's a whole lot of other tRNA right there, the chain. And this tRNAs form a chain. Like it's longer than this, but anyways, this chain is the first form of protein, also known as the polypeptide. Oh, also, the ribosome is also located right here, and it helps the base pairs of the tRNA and mRNA connect together like well, because if the adenine fuse with like guanine, it becomes a real issue. So it helps the base pairs connect each other well. This phase is called the translation, and this is basically it for protein synthesis. Transcription, regulation, and translation. All these basic steps, after these basic steps, the first form of protein is formed. But actually, this is not the complete form. It is just the first form. Now what you see right here are the steps for a protein to become a complete and well-functioning state. First, the primary. The primary is the polypeptide, of course. It's just the initial state of the protein. Secondary, the polypeptide is like an evolving stage for the polypeptide. It's not that important, secondary. It's just like a stage that is getting ready for the full functioning protein. And this tertiary phase is the complete state of the protein. Almost all of the protein in our body is in a tertiary state. And the quaternary. Well, the quaternary isn't necessary because it requires several proteins, more than one proteins to function well. Yeah, it requires more than one proteins. So in this state, the proteins work together to function well. You just have to remember that the tertiary phase is the most complete and well-functioning state of the protein. And that's actually it for protein synthesis. So let's review what we talked about. Protein synthesis is divided into 3 stages: transcription, regulation, and translation. First of all, the mRNA is made from the DNA in the nucleus and then travels through the ER to the cytoplasm, and that's the end of transcription. And in regulation, in the cytoplasm, the mRNA is divided into parts that is necessary for the protein synthesis. And the parts that are not needed for protein synthesis is called introns, and the part that is needed is called exons. And these parts of exons connect together to form a new mRNA strand. Finally, in the translation phase, the mRNA strand bonds, connects with tRNA represented as codons, and with the support of ribosomes, it forms an amino acid chain known as the polypeptide, the first form of proteins. And here, as the polypeptide evolves and develops new functions and its shapes, it becomes a final full functioning protein called protein state called the tertiary state. That is all for today. Next time we are going to learn about the roles of the nucleus and its specific functions and the little nucleolus that is inside the nucleus. Thank you for listening.